The effect of Porcellio scaber on the growth of Raphanus raphanistrum, performed by Bethany DePoy, Lindsey Hensley, Taylor Metzler, and Erika Ramirez. We did our project observing whether or not Porcellio scaber, also known as woodlouse, have an effect on the growth of Raphanus raphanistrum, commonly known as radishes.
We did our initial research on the terrestrial isopod Armadillidium vulgare, commonly known as pillbugs. Pillbugs exhibit worldwide distribution according to an article by Gerard published in 2013. Pillbugs can be found in any area that has humidity, including compost piles, leaf litter, beneath logs, and among rubble piles as identified in Wright's article in 1997. In many terrestrial ecosystems, isopods play a key role in decomposition via the breakdown of plant litter and by enhancing microbial activity. Pillbugs are classified as detritivores and consume dead plant material, then release it back into the environment in the form of feces, according to an article by Wood in 2012. Both of these processes performed by isopods contribute to nutrient recycling, as stated in the article performed by Albelho and Moles in 2009. An article done by Holland in 2014 stated that pillbugs encourage soil microbe activity and thus increase soil quality and contribute towards overall agricultural success, which is what we decided to investigate. Since we did not have access to A. vulgare, we decided to evaluate the connection between a functionally similar isopod, the woodlouse, and its effect on plant growth. We hypothesized that a larger amount of woodlouse will greatly enable the growth of radish plants.
The experiment began by setting up 8 glass fish tanks that were divided into 3 sections. Into each divided section, we added equal masses of dirt and leaf litter that were collected from North River in Bridgewater. 4 radish seeds were placed into each of the 24 sections. The first 6 sections received no woodlouse and therefore were the control. The remaining 18 sections were divided into 3 treatment groups, with 6 sections in the tanks receiving 3, 10, and 17 woodlouse. In order to keep the woodlouse contained but able to breathe, netting was placed on top of each tank, held in place with tape. This allowed oxygen and airflow for the woodlouse. The tanks were placed in the greenhouse throughout the duration of the experiment. Watering was done on an as-needed basis as to not overwater and drown the woodlouse. We watered each tank with 25 milliliters of water using the same 100 milliliter beaker. The soil was constantly kept damp in order for the radishes to grow. After 30 days of growth, the radishes were removed from the soil. The number of leaves were counted on each plant, excluding the cotyledons. The height of each plant was measured from the tallest leaf to where the roots began. The radishes were then dehydrated by placing them in a drying oven and recording their biomass with an analytical balance to get an accurate measurement. The woodlouse were also removed from the tank and counted, sorting the live ones from the dead.
The first set of data was not supportive of our hypothesis, thus resulting in inconclusive data, as the highest average height was observed in tanks with 10 woodlouse. If the data supported our hypothesis, then we would have seen the highest average height observed in tanks with 17 woodlouse and the lowest average height in the control tanks. The second set of data was observed to have a positive trend, with treatment groups in 0, 3, and 10 increasing in average weight with more woodlouse present. However, this trend was discontinued in treatment group N=17 as the average weight decreased. Our third graph supports our hypothesis. The data collected observed the average number of leaves on the radish plants increased as the number of woodlouse increased. Therefore, the smallest number of leaves were observed in the control tanks, while the largest number of leaves were observed in the tanks with the largest amount of woodlouse, which was 17 isopods. The averages for the treatment group N=17 was only out of 5 tanks instead of 6, since there was no plant growth in tank 24. Limitations within our project include not having the isopod of choice to collect data from, isopods getting stuck to the tape that was placed inside the tanks to keep the netting in place, and not having plant growth in tank 24 due to human error. These issues would be addressed if the project was repeated. Relating back to the work done by Holland in 2014, our experiment did show that the treatment group of N=10 woodlouse encouraged soil microbe activity and led to overall more plant growth compared to the control.